Good afternoon, everyone, to the second GoEQIPD webinar from our webinar series. Today, we welcome Kim Wever from Radboud University Medical Center in Nijmegen. And Kim is, a long time EQIPD veteran, been there from the beginning, and also is now one of the guarantors of EQIPD. And is also an assistant professor in anesthesiology, where she's heading the meta-research group. And today we hear a presentation from her entitled Systematic Review and Meta-Analysis of Animal Studies. Kim, welcome, and the floor is yours.
Thanks, Renee. I will hereby start sharing my screen. I hope that you can all see it. Oh, sorry, go back to the first slide, please. Yes. All right, so welcome again, everyone. Renee gave me already a nice introduction of the topic and who I am. But still, I'd like to inform you a little bit more. First of all, with the statement that I have no financial conflicts of interest. And I am indeed an assistant professor with the meta-research team at Radboud University, and I'm heading that actually together with my colleague Carlijn Hoijmans, who you can see next to me in this picture. By training, I'm a biologist, and I spent my PhD and part of my postdoc performing in vivo research in rodents mainly, some pigs. And I moved since 2012 into the meta-research field of preclinical studies, performing my first systematic review with Carlijn at that time, and developing a lot of methodology because so much was still not known about how to perform systematic reviews and meta-analysis of animal research correctly. Nowadays, I train people many times in how to do their own systematic review. We are members of CAMRADIS, the international collaboration of preclinical meta-research experts, the evidence-based toxicology collaboration, and also GoEQIPD, of course. And I'm a co-founder and admin of Prospero for Animals and preclinicaltrials.eu. And if you want to reach me or us, here's how to do it. So I'm really happy to speak to you today about preclinical systematic reviews, because as Malcolm spoke last week, and Thomas as well, on the causes and origins of translational failure from animal studies into clinical benefits, systematic reviews have been very instrumental in showing us where these pitfalls or at the moment still incompleteness of animal research are. And I'd like to show you today how systematic reviews have helped us discover these flaws and what they can do for you, maybe in your field, and what their benefits are. And if you're still not really familiar with what preclinical systematic reviews are, I'm now first going to show you a video scribe that we made a couple of years ago that explains the concepts. And it takes about 2 minutes to view. So I hope that you can see and also hear the sound.
Although controversial, animal studies are still an essential step in the development of therapies and drugs for humans. This is based on our belief that the treatment efficacy and safety found in animals will translate to the patients we intend to cure. But what if translation fails and the promising results obtained in animal studies fail to translate to patients? Despite successful preclinical testing, up to 85% of early clinical trials for novel drugs fail. When animal studies do not predict the response in patients, laboratory animals may have been used ineffectively or unnecessarily, and patient safety may be at stake. Ethically, as well as economically, such research waste is unacceptable. So what determines the translational value of an animal study? Which factors influence treatment efficacy in animal studies? A thorough assessment of the available preclinical evidence may provide vital information to optimize future research and improve translation from animals to patients. But with hundreds of new animal studies published every day, it seems impossible to read up on all the literature. We urgently need methods to obtain a structured overview, and systematic reviews are up for the job. A systematic review is a literature review focused on a single specific question, such as, Does fish oil improve memory in animal models of Alzheimer's disease? It aims to identify all available studies related to this question and to assess the quality of the evidence presented. It can synthesize new high-quality evidence from existing data. In fact, systematic reviews are regularly used in evidence-based medicine, where they are regarded as the highest level of evidence. But this methodology is not yet common practice for animal studies. A systematic review of animal studies is comprised of 6 main steps. Phrase the preclinical research question. Search for all evidence related to this question. Select the relevant animal studies and extract study characteristics. Assess the study quality and summarize the evidence. If possible, perform a meta-analysis, a statistical method to synthesize new evidence from existing data. At the top of these steps, interpret your results and enjoy the overview. So remember, systematic reviews of animal studies provide an overview of all available evidence on a specific topic and can point out knowledge gaps. The quality of the available evidence is appraised critically. Using meta-analysis, we can identify study characteristics which influence treatment efficacy. Their outcome can be used to inform the experimental design of future animal and clinical studies, and can help you choose the best animal model for your study. Systematic reviews can provide valuable insights into the causes of translational failure, so future research may be set up for success.
Okay, so I hope that was a good first overview for you about what systematic reviews of animal studies are and how we tend to use them. So here is a very brief recap again of the definitions. So a systematic review is the whole process of locating, appraising, synthesizing evidence from scientific studies to obtain an overview, and meta-analysis can be an optional part of this. So I will speak first a bit about the other steps of the systematic review process and later on specifically about meta-analysis and what it can do. So a short history of systematic reviews is that it all started in the clinical field. So in evidence-based medicine, as mentioned in the video scribe, the Cochrane collaboration is of course a driving collaboration that we, most of us are maybe familiar with, named after Archie Cochrane, who was one of the first who said that we should do an assessment of all available evidence to inform our future research questions and to synthesize evidence rather than try to figure out things by looking at individual studies one by one. But for animal studies, it's still relatively new. We found that the first systematic review of animal studies was actually performed in the Netherlands by Janneke Hoorn in 2001. And she was a pioneer here, and a couple of years later, that was followed up with the founding of Camarades in 2004 by Malcolm MacLeod and David Howells, joined by Emily Senna and others. In 2008, SYRCLE was founded. It was the Center of Expertise for Systematic Reviews of Animal Studies in the Netherlands, where me and Carlijn used to work until 1.5 years ago. And still other initiatives are evolving, like BRISA, the Brazilian Center of Expertise that was launched in 2021. And it's now becoming more and more common to see systematic reviews of preclinical studies in veterinary science, toxicology, etc. So it's a developing field, but relative to the amount of animal science evidence produced every day, we are still only reviewing the tip of the iceberg. So if you want to join in, that's still very much possible. So here is a video scribe screenshots where I display again the 5 benefits of preclinical systematic reviews. And I'd like to show you more examples of what these benefits look like in practice. So let's start with the first one, and that is how systematic reviews can provide an overview of all available evidence. And I will also give you an insight in how that works for evidence bases of different sizes. So here, for instance, you see a characteristics table, which is the main key element produced in a systematic review that gives you insight in the types of available evidence and also the amount. This table, in my opinion, is the most important part, the most important output of a systematic review. And here it looks quite comprehensive because it's from a review of melatonin as a treatment for stroke, so in animal models of stroke. And it was made by Malcolm Cloud already some years ago. And there were at the time only 13 studies that had tried to use melatonin as a treatment for stroke in animals. So with 13 studies, you can get this nice overview where you display the key characteristics of each study. Of course, which study it is, then the sex of the animals, how many experimental and control animals there were, the dose range of melatonin given, the time to treatment, and so on and so forth. So from this, it's already very informative for all the readers to see which evidence is already out there and which models are in use. So depending on what your review question is and how broad it is, your characteristics table might become a little different if you have a larger evidence base. So here is an example from a review by Gillian Curry, and she wanted to review all the available models for bone cancer pain. And there were a lot more. Over 130 studies were included in her review. So in this characteristics table, you actually don't see the individual articles anymore, but you see which cell lines are used to induce the cancer in mice versus rats, which strain of mice was used, again, the sex of the animals in each of these conditions, the injection site to induce the bone cancer, etc. So in her reference list, of course, you can find which of these 130 studies go where, but again, you try to make it insightful to the reader which animal models they could, for instance, consider if they wanted to use them to study bone cancer pain. You could take it one step further, and that is what me and Simon Yao did at some point in our lives when we felt it was a good idea. We wanted to evaluate all the animal models used for the treatment of intestinal anastomotic leakage. And there were a lot of them. There were over 1,300 studies on this topic at the time. That's quite a lot. I would advise to reflect before you embark on such an endeavor. But it had some really nice outputs in the end because the characteristics table takes also the form of a timeline of this field. Intestinal anastomosis field is quite old. It dates back to before 1970s. And here, for instance, is a graph depicting which species are used in which percentage of the studies from the 70s up to 2014. And you can see a lot of interesting trends happening. For instance, when the field first started, dogs were the dominant species that was being used. But over time, maybe due to ethical considerations as well, rats became the dominant animal model to study intestinal anastomotic leakage. And you can also see, for instance, here in this panel, which factors were introduced to make the anastomosis leak. So there's a lot of variation there. At some point in time, there was more focus on chemotherapy or peritonitis and sepsis. So if you have a lot of studies, you can also follow trends in your research field over time. And that was really exciting about this project. Having an overview of all available evidence also leads us immediately to the next benefit, which is identifying knowledge gaps. Because if you know what evidence exists, it will also be much easier to know which evidence is lacking. So an example of this can be seen here again. If we go back to the melatonin in stroke characteristics table, you can easily identify from this table a number of areas where there is a lack of knowledge. For instance, here is the gender or sex column, and it becomes apparent from this characteristics table that there is a lot of sex bias in this field. All the studies used male animals, except for 2 that didn't mention which sex the animals had. So there's a clear knowledge gap here regarding the effect of melatonin in stroke in females. And that's clinically relevant as well, because of course women get strokes as well as men. Another knowledge gap that I can point out here is, for instance, the time to treatment. You can see that many of these studies, actually all of them except for 1, maybe 2, focus on prophylactic treatment of the stroke, which is a bit of an issue, again, when you try to translate this to the clinic, because people would normally not come into the ER before their stroke happens, but after. So here, this column tells us that almost everyone is giving the melatonin either before or simultaneously at the time of stroke induction. And there's a clear knowledge gap about what the efficacy of melatonin could still be if you apply that after stroke. There's only 1 study that is really studying that. This one is infusing the melatonin over 2 hours. That's not really what would happen in a clinical setting either. Actually, if I show this table to different audiences, they will also point out that, for instance, here almost all the models have temporary ischemia. Some of the routes of drug delivery are maybe not what you would do in patients, etc. So having an overview of all available evidence gives you really good pointers of where you can base your next research. What are the gaps that need to be addressed? So that brings us to the third benefit of preclinical systematic reviews, and that is the critical appraisal of the quality of the evidence that we have. So it's always an essential step in a systematic review. Many narrative reviews, of course, give a really good expert opinion of research in the field, but an official critical appraisal that's systematically performed by several reviewers is usually not part of that process, but we always do it in a systematic review. So we can actually have 2 layers of critical appraisal. One can focus on the reporting of study quality indicators. So you look into the reporting of, for instance, any mention of blinding. Was it mentioned that anything was blinded in this experiment? Or was anything randomized? It could be selection of the animals or how to give the slides to the pathologists. Any mention of a sample size calculation, mention of regulation of the animal's temperature during surgery, that was important in this particular review, ethical approval or conflict of interest statement. So you're mainly here focusing on reporting. These data are from a review of mine on myocardial infarction models. And you can see that many of these reporting quality indicators are not reported. So that's unfortunate. This is, for instance, one of the points that ARRIVE guidelines try to address to get this graph to look more green, so to say. Yeah, succeeding in that, well, again, with Simon Yao, I could create a timeline for the 1300 anastomotic leakage studies. From starting from the 70s, the percentage of randomized studies, at least this study mentioned the term randomization. For blinding, it's going a bit slower. Blinding of the secondary outcome is coming a bit into view here in the recent decades. That's mostly histology that's blinded. Primary outcome, not yet so much. But in other fields like the stroke field, these improvements in reporting have become more apparent, fortunately. But apart from reporting, we also want to do something more in a systematic review. We want to perform an actual assessment of the internal validity of the studies, so a risk of bias assessment. And for that end, we have developed the SYRCLE risk of bias tool for animal studies, akin to Cochrane's risk of bias tool for human studies. That tool addresses several types of bias in controlled animal studies in several bias domains. And we have extensive guidance that authors can use how to assist them in how to make their judgment. So for instance, here you see the first type of bias that we try to assess, that is selection bias. It has 3 domains. It can occur during sequence generation, so that has to do with randomization. Baseline characteristics could still be different, even though you try to randomize. And allocation concealment is related to blinding at the time of selection. And then here are the signaling questions that you can use to assess the risks of bias in each of these domains. For instance, was the randomization sequence adequately generated and applied? So how are we faring in those assessments? Well, not yet optimally. Here is an example again from my review on myocardial infarction. Out of 500 studies, you can see that most of the risks of bias are unclear. That is a direct consequence of the fact that reporting is often insufficient. So, for instance, for random group allocation, the word random might be mentioned, but we have no idea if that means picking animals randomly from a box or actually using a random sequence generated by the computer to allocate the animals. Similar baseline characteristics are one of the highlights that are a bit better, but on all the other risks of bias, we still have concerns where the risks are unclear. They might be low, but they also might be high. But at least systematic review methodology makes this insightful so that you can give recommendations to the field on how to improve and also adequately account for these risks of bias in your conclusions, for instance, from your meta-analysis or your descriptive synthesis. So that brings me to two factors, benefits of systematic reviews that are related to meta-analysis, and that is how to identify factors that influence treatment efficacy and how to inform the experimental design of future studies. For the last one, we've already seen, of course, some examples because we know where knowledge gaps are and how to improve the quality of the studies, but there are also other aspects that we can find out through meta-analysis. So let's have a look at how that's done. So if you're new to meta-analysis, that is an analysis of analyses. That means that you're going to statistically combine the data from primary studies. So those studies have reported probably aggregated data, and we're going to statistically combine them. Why would you do that? So the aims of a meta-analysis of animal study data are to evaluate the efficacy of the intervention, or if it's in toxicology, for instance, to evaluate if there is harm. We focus for preclinical studies on the direction of the effect, so whether there's a benefit, a neutral effect, or a harm. Not so much yet on the precise estimate of the effect. But one thing that is quite unique to animal meta-analysis is that we can explore heterogeneity to generate new hypotheses, and I will show you some examples of that in a moment. And with these new hypotheses, we can guide the design of future the trials in animals or in humans, even if we decide that we're ready for a clinical phase. And we find new results without using new animals. So we first make the most of the data that we already have. And that is, from an ethical viewpoint, of course, a very good idea, as well as to prevent research waste. So here is an example of a meta-analysis. If you're new to that, the backbone of that is the forest plot. That is the plot over here. I will show you a couple more. But what you can see is that you start out here on the left-hand side with your individual studies. They all have reported data on a certain outcome. In this case, the left ventricular ejection fraction in the heart, in means, standard deviations, and the number of animals in the group. In this case, this is data from metformin-treated animals and control animals. But what eventually will happen is that you plot the effect of metformin on myocardial infarctions found in each of these studies in the forest plot. So you can see how variable these effects already are amongst each other, and also what the variance is within the study. And in the end, of course, what we like most as meta-analysis is this diamond over here that represents the pooled effect. So if I statistically pool the results of all these studies together, what do they collectively tell me about the effect of metformin? Well, in this case, they tell me that metformin increases your left ventricular ejection fraction, so that's good for your heart. We know that because here is the line of 0 that represents no effect, and this diamond is all the way to the right. So this is a positive effect of metformin on the ejection fraction. That's just an example to help you understand my next slides. Oh, I have one more. Another option is that you would have some outcome measured in events, for instance, deaths out of total. In this case, 15 out of 27 animals dying in the control group, and these numbers in the metformin group. So then you get a slightly different buildup of your graph, but you can do that for a lot of different types of data. That's the main point. And how do we interpret and go forward with these results? Well, the most interesting part is in subgroup analysis. So after we identify, for instance, that a drug class, that interventions as a whole have a positive effect, we can identify which drug classes are the most promising. So here's an example of how that could work. This is a review by Sarah McCann on antidepressants as a treatment for stroke. And she first grouped all the antidepressants together and analyzed whether they had an effect on the neurobehavioral score of the animals. So that's basically what their behavior is, or their performance is in neurobehavioral tests. Here you can see that effect size and the overall effect. So the diamond that I showed you in the previous slide is represented now by this gray bar. So that's a bit of a different representation. It's on the side. This gray bar is the effect of all the different drug classes together. They improved the neurobehavioral score by over 50%. So apparently all these antidepressants are quite effective in animals in improving brain function after stroke. And then she performed a subgroup analysis where you make subgroups of all the different classes of antidepressants. So what you basically do is you create separate diamonds for each drug class. Here they are plotted as bars. But what you can now see is that, for instance, the MAO-B inhibitors, they don't seem that effective at the moment, and there's a lot of variation in the study results. Lithium is quite effective. TCA also looks maybe promising, but here are the ones that look most promising. And this one is actually performing even better than the overall effect. So based on this subgroup analysis, you can select a group of drugs which you think is the most promising to take forward to future experiments in animals or in humans. So drug classes is one factor that, of course, could determine the efficacy of the treatment. There are also others that you can investigate in a similar way. One is, for instance, the effect of the study quality. So going back to our quality assessment, here is clear evidence that if you assess whether in meta-analysis studies that have not blinded the results are doing better than the ones that have blinded the results, and here you can see that that's the case. So this is a study about infarct volume in stroke after some treatment. Again, the gray bar is the overall effect of all studies together. And then in a subgroup of not blinded versus blinded studies, you can clearly see that not blinding the study tends to lead to an overestimation of how effective the treatment is. So the treatment appears more effective if you don't blind. And also Sarah, in her article, created a quality score, and she looked at the regression of that score in the with the effect size, and you can see that as the quality score goes up, the effect size that is found in those studies goes down. So missing out on certain study quality indicators tends to lead you to overestimate how effective treatment is. And that emphasizes the purpose of the quality assessment as well. Here's another example that I wanted to show you. Maybe you're really focusing on designing your next preclinical experiment, then this might be of interest to you. There might be even differences between rat strains that you would never actually be able to assess without meta-analysis. This is an example from Joris van Drongelen. He was interested in vascular function during pregnancy. And he looked at studies that used vasodilators and vasoconstrictors to influence the vessels of pregnant rats, and looked at how reactive the vessels were to those chemicals. First of all, I'd like to point out here that he compared the Wistar rat with the Sprague-Dawley rat in 3 different stages of gestation. And for early gestation, there is actually a knowledge gap because not many people are studying early gestation at all. So this also gives you again information that if problems with the vascular function in pregnancy happen already early, we don't really know that yet. Mid gestation and late gestation, there is more data. And here in late gestation, you can see the differences between the rat strains popping up. So the Sprague-Dawley rat is responding to vasodilators and vasoconstrictors the way you would expect them to. So this is healthy vascular function. But the Wistar rats tend to not respond anymore so much, at least for these drugs here. So their phenotype represents more a complicated pregnancy, for instance, in a patient with hypertension or preeclampsia. And that was very interesting to Joris because he wanted to model preeclampsia at some point and thought that the Wistar rat would then be the best rat strain to go forward with. And since there are hardly any studies directly comparing the Wistar with the Sprague-Dawley, he would never have been able to observe this from the individual studies. He could only get to these results using subgroup analysis. Then last thing that I wanted to show you is how we can directly inform clinical trials with meta-analysis of animal studies. That happened for the EuroHype trial. It was a trial that was planned to cool patients that come into the ER with a stroke. Of course, it's hard to pilot such a trial in terms of how long you should cool the patient, to what depth you should cool the patient, for how long the stroke might have been going on until it's still effective. So what the group of Bart van der Worp et al. Did, together with McLeod and Senna as well, was that they first summarized and synthesized in a meta-analysis all the animal studies on this topic, and they looked at the factors determining the cooling, the hypothermia protocol. So the time until treatment from the start of the stroke, the depth of the hypothermia, the duration, the timing, etc. And from each of these subgroup analyses, they picked the most promising condition in animals and took that forward to the design of their clinical trial. So when it's not really possible or ethical to test such a protocol in humans, animal meta-analysis data can actually be the only basis that you can have to develop such a protocol. Then I'm making a small sidestep to another very important function of meta-analysis, and that has to do with publication bias. So with all of these meta-analytical outputs, you have to take into account if they are maybe at risk of publication bias. You're probably familiar with the concept. So that's the fact that the likelihood of publication is lower for neutral or negative results, or results based on small sample sizes, or results that contradict what is currently thought to be true about certain intervention, for instance. The problem with that is that it can distort your meta-analysis results. So you aim with a systematic review to identify all available evidence, but if part of that evidence is not published because of publication bias, then you can have an overestimation or an underestimation in your meta-analysis of the treatment effect. And that can have a large impact on the conclusions that you draw. So all these analyses that I've showed you go hand in hand with an assessment of publication bias. How would we usually do it? One of the key plots that we use is the funnel plot. So here we plot the data points in the systematic review, or the meta-analysis, sorry, the individual studies a bit differently. We plot them according to their effect size and their precision, in this case, the standard error. The plot should take a symmetrical form, like a sort of a pyramid around the pooled effect. So this dotted line here represents the pooled effect. In this meta-analysis, the pooled effect of intervention is about 3, and the dots are nicely spread around this axis. Here below, on the lower side of the plot, are the small studies that have large standard errors. Here at the top are the precise studies that have a very small standard error. But what you actually often see is that the plot doesn't look like nicely symmetrical like this, but it looks asymmetrical like this. So on one side of the graph, some studies are missing, and that are usually the studies that have a small sample size and a neutral effect, or an effect size that contradicts current beliefs. And we try to assess whether those missing studies that haven't been published have a large impact on the output of our meta-analysis. So we can do that using trim and fill analysis. This has taught us a lot about how many studies are missing from the evidence that we can find in, for instance, PubMed. Here's an example of that. So again, you see this plot. That's an output of a systematic review by Sarah McCann, again, I think. Rather than eyeballing to see if this dot, this cloud of dots is symmetrical, we can assess that statistically. So we ask the software to tell us whether there are any access points on one side of the graph. In this case here, you see the software is trimming away a lot of studies. That is why this analysis is called trim and fill. And if these studies are superfluous on this side of the plot, that technically means that their statistical counterparts on this part of the plot have not been published, and they should exist, because the chance of underestimating versus overestimating the effect size are the same. And then in the second phase, you ask the software to fill the missing counterparts and put the trimmed studies back, and that will give you an adjustment of what the effect size of your meta-analysis would have been if publication bias had not happened. So if these neutral studies had in fact been published. This is a high-level overview, so I don't understand you to grasp how this exactly works right now. But it's important to know that this method has told us a lot about how severe publication bias is and how to adjust the effect sizes that we find in animal research accordingly, so that we don't overestimate effects because we actually are ignoring the neutral and negative studies. So that concludes my discussion of the benefits of preclinical systematic reviews. You might be feeling inspired to try this yourself, but where to start? Well, fortunately, we have a lot of guidance and methodology developed to help you out. So here's a picture of one of my favorite movies. Let's get help. Don't start by yourself because you will be reinventing the wheel. For each phase of the systematic review that you can see visualized here, there are different tools. So you start by phrasing the research question and writing a protocol for your review. You can do that with a publication that we had on the protocol format, and you can register that in Prospero for animals. Then you search for studies. For that, we recommend the step-by-step guide for searching that takes you through the whole process, and search filters for animal studies so that you don't have to screen through all the in vitro studies and clinical studies in your field. Then you come to the phase of selecting studies, inclusion, exclusion, doing that with multiple reviewers in a random and blind fashion, and extracting data, so annotating the species and the sex, etc., for the characteristics. You could do that in an online screening tool like SURF that's been developed by the Camarade's collaboration. Then addressing the study quality, SYRCLE'S risk of bias tool comes in there. I've shown it to you already. And if you have the availability to do meta-analysis and your data are suitable for meta-analysis, you can use the guides to preclinical meta-analysis to help you with the statistics of that. And in the end, if you discuss and draw conclusions about your findings, you might want to use preclinical grade. That's in analogy to the clinical grade, but adapted for animal studies. All of these steps are also discussed with the tools in the Camarade's Berlin SR wiki that you can find. There's also a database where you can see if any review on your topic or in your field already exists. There's even more, but I don't have time to go into all of these resources, but you can also find them on the Radboud UMC Meta Research Team website or the Camarade's website. And then, of course, there's also educational resources next to the published ones. For instance, SYRCLE'S e-learning on systematic reviews, the equipped e-learning course that contains SYRCLE e-learning in the module of systematic reviews, but also other materials on systematic reviews. We, and especially our colleagues in Berlin, but also sometimes in Edinburgh, organize 1-day and 2-day workshops that take you through the process. There's also weekly consultation sessions by Camarade's Berlin. When you really get started and you have questions along the way, you can call them and they will help you out. We have personal coaching, especially for researchers in the Netherlands, if they happen to be in the room. And a lot of internships happen at our departments throughout the year. So a lot of resources there for you if you're interested in those. And I think that concludes my presentation. So I'd like to thank you for your attention. And I think I managed to reserve 50 minutes for questions, and I'm happy to hear from you. Thanks.
Thank you, Kim. That was a great presentation, great overview, and lots of resources that you've shown. So yeah, so with this, we'll open the floor for questions.
You could unmute yourself or post something in the chat.
Yeah.
Maybe some people are having some background noise where they are.
Okay, so let's give people a few minutes and I'll start with a question. So, I mean, the evidence is there that systematic reviews are informative if you have a certain study question, and would be, I don't know, really beneficial to know more about already during your education, during your PhD program, or while becoming an MD about it, and also to practice this. What is the current status in terms of curriculum development? I mean, you can obviously just speak for the Netherlands, but overall, what's your feeling? It seems to be sort of in Europe that you in Nijmegen and Berlin are the hotspots, but is this spreading? Can we expect more in the future?
Yeah, that's a really good question. It's a bit of a challenge to come up with an answer in general. I think the Netherlands had a very privileged position for a number of years, because there was a motion passed by our government that it would be mandatory to have education on systematic reviews of animal studies in each laboratory animal science course in the Netherlands. So that's why we developed the e-learning. So anyone who aspires to be certified to perform animal experiments in the Netherlands has to get at least an hour of education on systematic reviews as well. So we're targeting there the people that want to go into the lab and work with animals, so that they get inspired to form maybe a systematic review before they start. I teach myself a lot in biomedical sciences and medical sciences courses. The concept of systematic reviews is revisited a lot, but mainly for the clinical field, not so much specific for the preclinical field. We have a minor here in Nijmegen that's quite unique. It's 9 weeks only on translating animal science into clinical benefit, but we get around 20 students per year. So we're targeting a very small group there. So I think the application of knowledge about systematic reviews to preclinical studies could still be boosted. So I hope that many more institutes will take that into their curriculum, so that students already can learn about this.
So I see we have a question here from Sarah. Sarah, would you like to speak up and ask your question?
Yes, sure. Good afternoon, everyone. Yeah, so my question is about the effect size. So what does it exactly refer to? Is it, for example, some factors or symptoms that we measure in a specific disease, and then their improvement would be the effect size? Or, because I could not really get this part.
Mm-hmm.
Thank you.
Yes, you're welcome. It's the lecture was a bit too short to go into all the details of meta-analysis statistics, but I can reshare my screen for a moment to maybe revisit it. Oh, it keeps doing that. Here, so this is the first slide, I think, where I explained something about the effect size. So one thing that's key in meta-analysis is that in the articles that you are reading, you're probably reading about means and standard deviations in a group receiving a treatment, in this case metformin, and a group receiving a control like a vehicle or a placebo. So you get from your individual papers, for instance, a mean of 28.8 and a mean of 32.4. And then you want to pool all the data from all the papers together, but you can't pool these two separate groups without first creating one effect size. So what you first do is that, in this case, this is a mean difference. It's also different between various outcome measures, but you just subtract the two. You say, in this first study, what is actually the difference in the ventricular ejection fraction between control and treatment? Well, the difference is actually minus 3.6 in this study. So, and it has a confidence interval. It says something about the variation between the animals in this study. So once I know that this study has an effect between control and treatment of minus 3.6, I do that for all the studies. So this one has minus 3.6, this one minus 0.76. So I first unify the difference between control and treatment into one number, because if I have one number per study, then I can pool them all together. And then this is the pooled effect size, and that tells me in this case that the effect of the treatment is an improvement in the left ventricular ejection fraction, so how good the heart is pumping, basically, of 9.3%, because the fraction is a percentage. If these were data about blood pressure, this would be, for instance, 9 millimeters mercury. If it was heart rate, it would be 9 beats per minute or something like that. But that can change a lot depending on how the authors of the primary studies put their data into their paper. But what you're trying to do is express in one number how effective the treatment is. And then for the subgroup analysis, you're doing the same actually per subgroup. And then you say, oh, are these subgroups different? So do I see a difference between this subgroup here, the SNRIs, and this one here, the TCAs, and you look at, in this case, at the height of the bar and the confidence interval that goes with that. Does that help you with?
Yes, thank you very much. Now everything is clear. Thank you.
You're welcome.
Hi, Kimberly. I have a question. I'm Deborah. I'm working at Janssen Pharmaceutica in Belgium. I really love your government that they kind of put that mandatory these in the curriculum. Thinking about doing that at Janssen, actually, kind of entering that. Yeah. So I was wondering, because I really got a fascinating talk. I really got motivated. I already have some couple of questions that I really want to try out. But I was wondering if you know if any groups are doing that as a service, for instance, because I can imagine some questions might take up a whole bunch of time to do that. So do you know of groups that kind of do that together with you, if you would, you know, get that result even quicker into the community?
Do you mean in Belgium specifically?
Well, not specifically. You know, I think those things can you know, happen internationally, but kind of have that, yeah, something like a service or a, you know, a cooperation with people that are actually used to doing that. Of course, you can initiate, and I do see the e-learnings helping you out. But of course, you know, if it might go quicker with people that are used to that. I was wondering if, you know, some groups have had the idea, or well, let's start this up as a service, for instance.
And by service, you mean helping
really
people
have a
implement this
question.
methodology in their institute, or helping people that are already working with systematic reviews?
Actually helping out people that have a scientific question. You know, you have the hypothesis or you have your question, and then help out with a systematic review.
Right, yeah.
To people that are not used to doing that, right?
Exactly.
But
Yeah,
really
so
would benefit of having that, yeah.
That is something that we do as one of our core activities, I think. So we start with training. So we give a lot of workshops. Anyone can join them with a vague interest in the topic. And during the day, for instance, of the workshop, they go through the steps of the systematic review. Some people stop there because they just want to, for instance, be able to read a systematic review that they find in PubMed. Others get inspired to go on and apply this for their own research question. And then we provide supervision or coaching, or how you want to call it, where we just follow the people and guide them through the process. In the Netherlands, again, we are very lucky because we have support from the government, actually, who gives a small grant to the researcher of 2 months of their salary, and 20 to 80 hours of supervision are paid for us to help them. But that's a very unique construct, of course. For people outside of the Netherlands, sometimes they have some funding. For instance, they take it from their European grant or from something else, and we help them out as well. And the colleagues in Kameradi's Berlin and Edinburgh do similar things that I know. So yeah, that we really help them as intensively or as loosely, depending on their previous experience. For some people, it's their second systematic review, so it takes us less time. Some people really are a complete novice, so then we give them a lot of supervision. Is that along the lines what you envision?
Well, yeah, I guess it all depends time-wise and the time needed to actually come to the answer of your question that you had in mind. I guess it really depends on the depth and the detail that you need. And I was wondering, like, if you would be able to quantify the time needed to get to the end, right? Kind of, it's really, and you see all these steps, and you immediately want to dive in, right? But I just have it like, and you have all the IT and the system aids and probably some AI to that helps you out, right? But time-wise, is that feasible? You know, we regularly have new animal models to set up, new projects coming in and out, you know, changing like monthly. We have new projects, and we have all these new questions over and over. And I'm wondering, so this is very fascinating and interesting and should be done more, in my opinion. But the time it would take, I'm just wondering how fast that goes, kind of.
Yeah. Well, you're not the first person to ask me that, of course. This question
difficult
is asked,
question probably too. Well,
yeah, but this question is asked almost every time, but it's also very difficult to answer because it depends hugely on the broadness or the narrowness of your research question. So I showed you the example of 130 and 1300 studies. The 13 study review might be completed in 6 months from searching to publishing it. 130 studies, not so much. I think it would take you about a year. 1300 studies, don't try it without automation. Then maybe with automation, it could take a year if that runs smoothly. But it also depends on a lot of other things, even how many reviewers are working on this. In my experience, for instance, many of my people that I supervise are either PhDs or they are doctors in training to be specialists. The PhDs have a lot of time often and they can dedicate it, but the doctors have to put it in between their clinical activities. So it's a bit of a stop-go process and that makes it take much longer. So there's all of these factors that you can take into account. You can sort of estimate it up to the levels of screening, but after that it becomes more difficult to say, especially for data extraction, because some people want to do like 20 outcomes. They want the heart rate, the blood pressure, the ejection fraction, the size of the ventricle, and they're interested in everything. And some people just say, this is gold standard. We just want blood pressure, that's it. Yeah, that makes a huge difference. Even if you have 50 studies, how much data do you want to take from those studies?
Okay. Yeah, thank you.
Yeah, so we decided on a case-by-case basis, we can give some advice on it.
Yeah, I think I'll try that on a kind of a small, if that's possible, on a small scale, because we kind of theoretically we always do some kind of review, but that's kind of an Excel based, right? You kind of go to PubMed, you look for all these publications, and you try to keep up with what information is in there, and you have this tiny Excel. So that's a really small scale, but of course, and you miss a lot of publications that way, right? Because there's no automation in that. But yeah. I think I'll just try to see if at what scale I can do that myself. Yeah. Thanks a lot.
Just start small, baby, and then if it works for you, then scale up. Thanks.
Thanks.
Maybe I can ask one more question, because I mean, like you've shown us really good evidence in terms of why people should go into this, but also good reasons why not to do this. Because if it takes up a year of your research time before you start, but then you say like, well, this could be a year really well invested before you dive into something that you know sort of may not lead to a translatable outcome. So are funders picking up on this more, that they would, for larger projects, require systematic reviews before starting? You can comment on that.
Yes, I can comment, and my feeling is not yet. That there was a lot of hesitance or a lot of discussion as well between making things mandatory or making it an advice. The Dutch government also passed a motion that systematic reviews should be the norm for animal studies. But you can see the nuance there, because they don't want to mandate it, because that can also, sometimes it's maybe not necessary. Also then you're imposing something that's mandatory to people who have don't have the resources or the knowledge. Our main funder is ZonMW, that's the national funding agencies for biomedical and medical research in the Netherlands, and they are really supportive of the methodology. So they will put it, for instance, in their call text, like, we recommend that you do it, but it's not really mandatory, I think, in many funding agencies. I wouldn't be able to name one where that is the case. But so, yeah, picking up on it, yes, but yeah, cautiously maybe.
Yeah, no commitment yet. Okay. Yeah.
yeah, it would be great actually if there was even more attention for it, because I also, from Deborah's comments, I remember again, I wrote my last chapter of my thesis. No, I wrote the first chapter last. That was the systematic review. I reviewed the animal models that I was working with last, after I'd already done all my experiments, because I wasn't familiar with systematic review methodology when I started. Then I put it as the first chapter in my thesis, because I said, well, this is an excellent overview of all that has been done. And well, luckily we did something that was new, because we could have accidentally duplicated something, maybe, because you never know if you're missing any evidence. And I think that is such an eye-opener for nearly all PhD students that I supervise. If they happen to be in the early phase of their PhD, they're like, oh, this is great, I now have a good overview. And if they're already finished, they always say, oh, I should have done this before. So yeah, that's I think it would be great if funders and institutes themselves at least pitch the idea and plant the seed that this is maybe a really good starting point to inform your actual experiments in animals that you're planning to do.
I think these were some great final words to conclude this webinar. Nobody could have summarized it better. Yeah, we want to thank you again, Kim, for a great presentation. And yeah, great overview, great material, in depth, really. And yeah, come back in 2 weeks when I'm going to present actually on exploratory and confirmatory research design. So stay tuned, and also check out our YouTube channel, where also this presentation will be uploaded later. Okay, have a good afternoon. Thank you.
Thank you so much.